My name is Jaroslav Bendl. I'm assistant professor at Icahn School of Medicine at Mount Sinai, and I'm going to talk about the research that I'm doing there. But first, I would like to start with a few words about from where I came from. So basically, I grew up in the era predecessing .NET bubble, and at that time, the internet was still in diapers, but the newspapers were full of predictions about implications of rapidly evolving tech on our everyday life. So I wanted to be part of the tech revolution, so I started to code, and I found liking for that. And I had my heroes. Yeah, you can see some of them. Sorry for that. So on my educational path, I exchanged a lot of code for some titles. As you can see, I developed some computer games, some evolutionary algorithms. And at the end of the path, 2016, I published a couple of tools for prediction of the effect of mutations on human health. So that's my niche in computational biology. And those predictions were sufficient enough to get me a job at Mount Sinai. So this is my genome, so don't take photos. Actually, you can, because that's just a very small part. It's my genome and your genome is consisted of 3 billion of letters, amino nucleotides, which are on these notoriously known double helices in form of chromosome within the cell, actually within the cell nucleus, which is a small part of the cell, and each of you has about 100 trillion of cells in the body. So think about that. And this thing is actually part of the genome where you can see the definition of ApoE gene, which is a polypoprotein that is responsible for removing cholesterol from the bloodstream. And as you can see, the mutations on these two particular places determine your risk for Alzheimer's disease. As you can see, in my case, my risk is about 2 or 3 times higher than the risk of most of you sitting in this room, which is actually not that bad, because 2 or 3 of you, I think, based on the number of you, are actually in the category which will most likely develop AD. While we know the mechanisms for ApoE gene and for these two particular mutations, we have dozens of thousands of mutations for which we don't know the function yet. And we need some mechanism, automatic mechanism, how to find the connections and how to find the treatment. In this case, these mutations actually abrupt the function of ApoE gene, and ApoE gene is supposed to remove cholesterol from the blood, but in oligodendrocytes with these two mutations, if you have them, the cholesterol start to be deposited in the cell, which is a problem because oligodendrocytes are supposed to remove to promote electrical activity in the cell, in the neurons, and their malfunction can basically trigger the neurodegeneration. So we have undergone a rapid transformation in molecular biology, which has led to genome sequencing and improvement in molecular biology, and the techniques has made it affordable. These days, we can actually test the mutations in the human body within stem cell models. We can introduce mutations. And we can grow these stem cells to neuronal cells and apply a couple of functional omics techniques to test whether those mutations are actually what they are doing and whether the treatment could be effective. So, for instance, we can find out what's the effect of mutation on a 3D shape of the chromatin or gene expression or protein levels. But these predictions, most of the time, are pretty imperfect, meaning that you can come up with hypotheses, you try to code it, you write a lot of models to evaluate them, but at the end of the day, when you test it in the lab, it fails, which says one thing: the models that we are using are imperfect and we need to do better. There is a lot of statistical methods on background, as well as a lot of machine learning, but this is not enough as it is. So I will, if we can resolve these problems in the modeling, we can change medicine forever. And how to do that? So I will walk you through the example on Alzheimer's disease and your potential visit 15 years ago, visit in the doctor's office, now and in the future. So in the past, your chances were not really good, because like at the time when you when you experience the cognitive decline, the doctor could probably just like give you some treatment for comorbidities like insomnia or depression. However, there is no treatment for Alzheimer's disease yet, and it's actually surprising that up to these days genetics didn't do much for AD in general. However, what will happen in future? In future, we will have the models which will allow us to do tailor-made treatment, which means you will log to the genome bank, and you will see not just all of your mutations, which actually you can see them today if you subscribe for particular services, but also cellular models beyond those mutations, and you will be able to get cocktails of drugs that are specifically designed just for you automatically. And you will be able to even test them in the dish, because like if I scratch my hand, I can take some skin cells under my nail, and then I can grow them back to the stem cells, and I can reprogram them to neurons, and I can put it into the dish. I can put there also some cocktails of drugs, and I can see whether it works or not. I mean, in most cases, we will just rely on the predictions, but there is always option to test it. And yeah, so we need to do a lot of work on these background methods, but it's all undergoing, and that's something what I devoted the last decade of my life.